single-cell foundation models have rapidly matured. Models like scGPT, Geneformer, and scBERT now dominate transcriptomic pretraining, and they all share a core architectural choice: masked expression value prediction. You mask a subset of gene values, and the model learns to reconstruct them. This objective excels at capturing gene-to-gene dependencies and latent co-expression structure. But here's the critical limitation. Reconstructing individual genes does not automatically optimize the geometry of a whole cell representation. For downstream tasks like cell type annotation, dataset integration, or gene regulatory network inference, you need a cohesive, discriminative cell embedding. Masked reconstruction simply doesn't provide explicit supervision for that. This mismatch is exactly what CoCoS addresses. The authors introduce a contrastive pretraining framework that learns cell representations through complementary transcriptomic views, while retaining masked reconstruction as a foundational objective. Standard contrastive learning doesn't transfer directly to single-cell data, however. We'll walk through why and how CoCoS solves three domain-specific challenges: biologically valid positive pairs, gene identity shortcuts in negative sampling, and the precise timing of contrastive supervision. First, constructing valid positive pairs in single-cell pretraining is non-trivial. In vision, contrastive learning relies on augmentations like cropping or color jittering that alter low-level appearance while preserving object identity. In transcriptomics, perturbing expression values can fundamentally alter a cell's biological state or dynamic programs. If you change the values too aggressively, you destroy the identity you're trying to preserve. CoCoS avoids this by partitioning each cell into two complementary gene views based on co-expression structure. The framework builds an undirected weighted co-occurrence graph across the training corpus, where edge weights reflect how frequently two genes appear together in highly expressed profiles. It then constructs a bipartition using weighted degree ordering and count-based placement. Genes are processed in decreasing order of their summed co-occurrence weights, and each gene is assigned to the panel containing fewer of its already assigned neighbors. This pushes strongly co-expressing genes into opposite views. For any given cell, view A masks panel 1, and view B masks panel 2. Every gene value is observed in one view and hidden in the other. The two views remain anchored to the exact same measured cell, avoiding artificial expression shifts while exposing complementary biological evidence. But complementary views introduce a new vulnerability: the gene identity shortcut. Single-cell data is highly sparse, and different cells in a minibatch typically retain different subsets of gene identities. If you use standard in batch negatives, the contrastive loss can exploit gene set composition alone. The model might learn to distinguish a positive pair from a negative pair simply by checking which genes are present, completely ignoring the correspondence between gene identity and expression magnitude. CoCoS neutralizes this with expression-aware contrast set construction. Specifically, it generates identity-matched hard negatives. For a given anchor, the framework keeps the gene identities and masking pattern fixed, but permutes the expression values across those genes. The anchor and the negative now share identical gene sets, but have scrambled gene-to-value mappings. To tell them apart, the model is forced to learn actual expression patterns rather than technical composition artifacts. The contrast set combines these hard negatives with standard cross-view and same-view in batch examples, ensuring the embedding geometry is shaped by biological signal. Even with perfect positive and negative pairs, training dynamics matter. If you introduce contrastive supervision too early, the encoder hasn't yet learned stable gene dependencies through masked reconstruction. The model will latch onto trivial technical cues like library size, sparsity patterns, or batch effects to align the views. CoCoS addresses this with a competence-gated contrastive onset. Instead of a fixed epoch schedule, the framework uses a label-free sentinel cohort disjoint from training to monitor readiness. At regular intervals, it computes three metrics: gene program concordance, which measures the separation between paired and mismatched views relative to off-pair variation; a repertoire score, which tracks embedding diversity via the effective rank of the centered covariance of normalized embeddings; and the pooled masked value reconstruction error. Only when concordance stabilizes, diversity is preserved, and reconstruction error drops below a calibrated threshold for 3 consecutive probes does the gate open. The contrastive weight then ramps up smoothly using a sinusoidal schedule. Until that trigger, the model trains purely on masked reconstruction. Once activated, contrastive supervision gradually shapes the whole cell embedding, while reconstruction remains active on both views. It's a data-dependent curriculum that prevents premature alignment. The results validate this 3-part design. Under a unified 10-dataset cell-forward protocol, CoCoS GPC achieves a mean KNN accuracy of 74.82% and a linear probe accuracy of 78.85%, outperforming scGPT and ranking first among comparable methods across multiple datasets. The fixed identity negative diagnostic confirms the mechanism works. Starting from a shared checkpoint, a model without hard negatives maintains a large confidence gap between ordinary negatives and value-permuted negatives, but introducing 4 identity-matched negatives collapses that gap to near zero, proving the model no longer relies on gene set composition. The framework also transfers effectively to gene regulatory network inference. When evaluating frozen gene embeddings for edge prediction across 6 benchmark networks, CoCoS GPC records the highest mean AUROC and AUPRC point estimates among compared variants. While the exact top-performing variant shifts per network, the consistent top-tier performance across both cell-type annotation and GRN edge prediction confirms that properly bounded whole-cell contrastive supervision yields more discriminative, transferable representations than reconstruction alone. CoCoS doesn't replace masked language modeling for genomics, it augments it. By structuring complementary views through co-expression partitioning, eliminating identity shortcuts through value derangement, and gating contrastive loss behind a label-free readiness monitor, it bridges the gap between gene-level reconstruction and whole-cell representation learning. As single-cell datasets scale and downstream tasks demand more robust cellular understanding, models that actually optimize cell-level geometry will become essential. If you're working at the intersection of foundation models and transcriptomics, this framework provides a clear blueprint for the next generation of biological representation learning. Thanks for watching.